Hi all, and welcome to topic 2, water and plant cells. This follows on from topic 1, where we looked at plant cells in isolation, which was largely a revision, but we also tried to place them into the broader function of plants as a whole. This is the second time I've recorded this lecture. The first time I had to get rid of my recording because I was so enthusiastic in my delivery that the audio was horribly distorted, like some sort of heavy metal concert. So here I am recording it for the second time, and this time I'm going to attempt to temper my enthusiasm. But I have to say, without sounding completely ridiculous, that the interaction between water and plant cells and water and the plant is extraordinarily interesting. And we take it from this simple understanding of how and why water moves in and out of cells to how and why water moves in and out of plants, all the way through to how can we manipulate this process. What does it do to a plant when we do, and how can we take advantage of that? Anyone with even the simplest understanding of life knows that water is crucial for all organisms on Earth, and plants are no exception. We know that photosynthesis requires CO2. Plants convert gaseous CO2 into solid carbohydrates in the leaf in an extraordinary process of photosynthesis. But this process also requires water. Water is split and plays a vital role in the electron transport chain, which is the first step in photosynthesis, aside from the actual absorption of photons from the sun. Water plays an important role in stopping plants getting thirsty, in other words, preventing desiccation. So water is absorbed from the soil and transported through the plant to prevent cells from desiccating, in other words, dehydrating. And then plants with their cell walls, as we'll see in the next slide, use this water content to actually provide support to the plant themselves. A very even slight imbalance in the amount of water that plants have can cause gross malfunction. Some plants can handle very, very little desiccation or dehydration. Some plants, like our arid desert plants, can handle or survive with very, very little amounts of water. And plants, as a rule, doesn't matter if they're a water lily all the way through to a cactus, plays a delicate balancing act of absorbing water and losing water from the leaves. One of the big differences between plants and animals is the fact that the cell walls of plants has a cell wall composed of cellulose. Very few organisms on Earth have these cell walls outside the cell membrane. Bacteria have a cell wall of sorts, but fungi has probably the closest to a plant cell wall. However, it's not composed of cellulose, it's composed of chitin. These cell walls play a vital role, as I touched on before, because they provide a structure to the cell. Our animal cells just have a membrane which can form all sorts of different shapes, but provides very little support. That's why we have a skeleton. Plants, on the other hand, use these cell walls for support. At its simplest support, they just fill them with water, and it's the pressure of that water being inside the cell wall that actually gives strength to the plant. Plants can take this further by adding lignin to their cell walls to provide physical, solid support. This hydrostatic or water pressure within the cell is referred to as turgor or turgor pressure, and so we'll be using the term turgor pressure for the rest of this lecture and this lecture series. Water is one of the most abundant molecules on Earth, yet it can also be the most limiting, and it plays an extremely strong role in the life of plants, such that we can actually graph the growth of plants to the availability of water, and we see there's a very tight relationship. For example, on the left-hand side here, we see some classic research from the 1970s, where they monitored the amount of water being used by crops and related that to the amount of the yield in tonnes per hectare they got from those crops. And as you can see, there's a tight linear relationship over the amount of water they measured here between water use and plant growth. So the more water use that was used, the greater the plants grew. On the right-hand side, we see a slightly different graph, but measuring a very similar thing. Instead of actually measuring the amount of water the crops used, in its place on the x-axis here we have overall water availability through precipitation. On the left-hand side, instead of having crop yield in tonnes per hectare, we have productivity or yield in terms of dry mass per square metre per year. Right, so this is over time. We see that the graph on the right-hand side is of a different shape to the left-hand side. On the right-hand side, it's sigmoidal. On the left-hand side, it's linear. This may be for two reasons. Maybe the water levels that were measured in this left-hand study represent this linear phase of the sigmoidal curve. Or perhaps it just shows that not all rain that a soil receives actually makes its way to the plant, and this is a very likely explanation for this relationship. As there's less and less water, we see down the bottom here that it's harder and harder for the plant to access this water, and so we get a smaller degree of change in the amount of productivity. As the soil gets saturated with water, here represented by precipitation over about a metre per year, we see that plants can't actually make use of this extra water because they're actually physically at their maximum production, and so production levels out. Plants use huge amounts of water, however most of it isn't actually used for cellular function. Instead it just goes from the soil into the root zone, up through the xylem, and then out through the leaves through a process of transpiration. And this is vitally important because it not only provides water to the canopy, of course, but it also allows for transport of nutrients absorbed through the root zone up to the canopy. So it actually uses the water in this sense as a transportation device. So 97% of it's lost to the external environment. The remaining 3%, about 2%, is used in growth for filling up the cells and allowing them to elongate. And 1%, just 1%, is actually used in the photosynthetic reactions. Interestingly, this 1% is very similar to the amount of light which is used in photosynthesis compared to the amount of light that a leaf receives. So of the amount of light received by a plant, only about 1% of it is actually caught and used to provide the energy for photosynthesis. We're not going to go through the nitty-gritty of the structure of water here, but I'd like you to remind yourself by reading the appropriate sections in Chapter 3 of Tate's and Zeiger about the properties of water and what the structure of water provides or allows for in its behaviour. So I'd like you to remind yourself of the properties of water in terms of cohesion and adhesion. These properties are important, especially the hydrogen bonding of water is important because it allows for what we call the tensile strength of water, the strength water has in being pulled. So when water is bound together in a chain in the xylem, in a long continuous chain from the root zone up to the leaves, that chain of water can be pulled with a lot of pressure and it won't snap. In fact, it can withstand tensile strength in the region of negative 20 megapascals of negative tensile pressure without snapping or tension. When it does snap, we get what's called cavitation and we get air bubbles forming in the xylem, and this is extremely dangerous for a vine or any agricultural crop. And you'll be learning about this in some of the more applied physiology subjects later on. A classic example of thinking about positive hydrostatic pressure or positive water pressure compared to negative water pressure is thinking about a syringe. And here it is represented in the slide. So we have an air bubble inside the main shaft of a syringe. We cap the end of that syringe. If we pull on the end of the syringe, there is now a pulling force on that air bubble. If we push in the plunger, the water now forces itself on that air bubble. There's positive hydrostatic pressure. When the water is pulling on that air bubble, as we're pulling that syringe out, we get a negative hydrostatic pressure. That's called tension. When water fills up a plant cell, the pressure that is formed by stuffing all that water inside the confines of a cell wall provides positive hydrostatic pressure. Water, when it's in a xylem and is getting pulled up, the xylem is actually under negative hydrostatic pressure. It's being pulled. So let's explore these properties of water a little further. In particular, the concept of water potential. Water potential is all about the chemical energy of water. When we talk about chemical energy, we're talking about the free energy of a substance. And free energy really is the ability of that substance to perform an activity. And in physical terms, we call that work, which is the ability to essentially move something over a distance, or force times distance is the technical definition of work. And that's generally measured in joules per mole, right? Mole of substance. Right? The chemical potential of water, therefore, is the free energy of water. How much potential does it have to actually perform an activity? Right? So if we carry water and put it up in a tank 20 meters above the ground, all of a sudden that energy, that water, which was then just simply sitting on the ground, all of a sudden has a whole lot of energy, activity it can perform because it's up in the air. It's actually got gravitational potential. So if we open the tap, that water will then move from the tank down towards the ground again, and we can harness that energy, and we do harness that energy in the form of dams, etc. So we can increase the free energy of water by moving it, say vertically, but we can also decrease the energy of free energy of water by adding things to it. So water with the highest chemical potential by itself is pure water. It has, when it's in its purest form, it has the most free energy for activity. When it has solutes dissolved in it, that free energy is reduced and its chemical potential is reduced. Water will always flow from higher chemical potential to lower chemical potential. So it will always flow from a position where it has a lot of gravitational energy up high, and it will flow down to an area of low chemical potential. So if we put pure water and poured it into a jar full of salty water, that water will diffuse about that container to try and even out the chemical energy. It will go from that area of high chemical potential, which is the pure water that you're pouring in, to the lower chemical potential, which is that salty water. So we've talked about the effect of gravity, and that's easy for us to understand. As we lift it up, it increases in gravitational energy or gravitational potential, but other things act on it as well. The concentration of solutes, known as the solute potential. There is also the potential added to it from pressure. If we put water into a confined space, we increase the hydrostatic pressure it has. It wants to burst out. So we're increasing its water potential if we confine it into a space. If we pull on that liquid, we are decreasing that hydrostatic pressure, right? Because it wants to move, it's pulling on it. The energy level or water potential is always defined relative to a reference of pure water. So pure water at ambient temperature and normal atmospheric pressure is defined as 0. And everything is described in relation to that. So for example, if we have pure water in an open vessel, so it's got no hydrostatic pressure acting on it, it's down at ground level, so there's very little gravitational potential, and we add salt to it, the salt will decrease the water potential of that fluid. So let's look at this solute potential or osmotic potential, the effect of dissolved solutes on the water potential itself, or the free energy of the water. Adding solutes decreases this free energy of the water by diluting the energy itself. So the water molecules are further apart. It's decreasing its energy. We can look at this as an equation. So the solute potential, psi s, equals the gas constant, right? So that's just a constant, by the temperature, so temperature affects it, by the actual concentration of the solutes in the liquid as moles per litre, all multiplied by -1. So if we increase the solute concentration because of that negative sign, that means the solute potential drops, it becomes more negative. Likewise, if we increase the temperature, what do we get? We get a decrease in the solute potential by increasing temperature. So the warmer the temperature, the greater the negativity of the solute potential. Hydrostatic potential, positive pressure raises the potential. So the pushing pressure raises the water potential. Negative pressure, the pulling or the tension, drops the water potential. The hydrostatic pressure. Thus, the water potential. Positive hydrostatic pressure when inside the cell, as I said before, is known as turgor pressure. So when we fill a cell full of water and that force it acts on the cell provides a positive hydrostatic pressure, which will increase the water potential, make it less negative. If we pull on that water, we're providing a negative hydrostatic pressure, which will decrease the water potential. So, water in the xylem has negative hydrostatic pressure acting on it because it's being sucked up, like through a straw, and that will be decreasing the water potential of the liquid in the xylem. Let's look at this in terms of a make-believe cell, which we're going to extract from a plant and add to beakers. We've got one beaker on the left, which is full of pure water. We see that beaker has a hydrostatic pressure potential of 0 because it's an open beaker. If we close that beaker and push down to the top, we then get a positive pressure because that water is being confined into a smaller space. The solute potential is 0 because it's pure water, and therefore the water potential, because there's no potential from the pressure and no solute potential, that means the water potential is 0. We just exclude any potential due to gravity. On the right hand side, we have a beaker containing 0.1 mole of sucrose. So we've added solutes to that water. The hydrostatic pressure is 0 again because it's an open vessel. However, now because we've added solutes, we've increased the concentration of solutes. That means we'll have a negative solute potential. Therefore, we add the hydrostatic pressure to the pressure provided by the solute potential, and we get negative 0.244. So the water potential of a 0.1 mole of sucrose solution in an open vessel is negative 0.244 megapascals. All right. Now, let's pretend we have a beaker containing 0.1 mole of sucrose, just like that beaker before. All right, so it's just sitting there on the bench. And we have in our hand, or probably in a very tiny small pair of tweezers, a dehydrated cell, or a flaccid cell, which has no turgor pressure at all, so there's no force acting from inside the cell outside the cell. We have that in a pair of tweezers. Inside that cell is enough solute such that the liquid in the cytoplasm has the solute potential of negative 0.732 megapascals. Therefore, the water potential of that cell is the hydrostatic pressure added to the solute potential, hydrostatic pressure being 0, solute potential being negative 0.732. Combine those together, the overall water potential is negative 0.732 megapascals. Now we're going to add this cell into the beaker, which we know has a water potential of negative 0.244. So the water potential of the solution is greater or less negative than that of inside the cell. It has higher chemical potential than inside the cell. And we know water wants to move from high chemical potential to low chemical potential, and it will keep moving along that gradient until they're evened out. So when we put that cell in, the cell has two choices. Either it loses solutes from inside the cell and they move into the solution to even out the water potentials, or it takes on water. And because of the cell membrane, it can't lose solutes. So the cell has the solutes that it started with. It's got so many solutes that its solute potential is negative 0.732. We add that to the solution, that can't change because the solutes can't move in and out because of the cell membrane. The only thing that can move in and out is water. Because there's a higher chemical potential outside the cell, water will move inside the cell. And that's what happens. As it moves in, the turgor pressure within the cell increases. That water is now increasing in a confined space and causing pressure to build up on the inside of that cell wall. So all of a sudden, the hydrostatic pressure inside the cell goes from 0 and starts becoming positive. And it starts, more water moves in and moves in and moves in. This pressure builds up to such a point that when we add the negative solute potential to the positive hydrostatic potential, it creates a water potential inside the cell which is equal to that of the solution, and therefore the water stops moving. So our cell expands again. Now let's take that cell, pull it out of that liquid. Now remember, it's got the overall water potential of negative 0.244, which is a composite of a solute potential of negative 0.732 and a hydrostatic pressure of positive 0.488. And we put that into a 0.3 molar sucrose solution. 0.3 molar sucrose solution has a 3 times lower water potential than a 0.1 molar sucrose solution. So its water potential is negative 0.732. So if we put that cell in, all of a sudden, water inside the cell has a higher chemical potential than that outside the cell. And what does water want to do? It wants to go from high to low. And so what's going to happen is that water is going to move from inside the cell to outside the cell, causing dehydration of the cell until the water potential inside the cell is equal to outside. And here we see what happens when we have dehydration of the cell membrane shrinks in on itself. So in herbaceous and small grasses, herbaceous plants and small grasses, water potential in the leaves is generally between negative 0.2 and negative 1. In trees, it's on average around negative 0.25 in trees and shrubs. They require a lower water potential in the leaves because they've got to drag, they need a greater gradient of water potential to drag water up from the roots. But we'll go on to that in a later topic. Plants in arid climates, their water potential can get down to negative 10 in the leaves. Those really watery plants like lettuce, cucumber and aloe vera can have a solute potential as high as negative 0.5. So very bordering on 0. So they've got very low solute concentration in the cells. Because of that, the tensile, the pulling pressure, because of the high solute concentration in the xylem and the cell walls, is all due to the hydrostatic pressure, they can actually get into positive territory. Plants wilt when their hydrostatic water potential drops from these levels between 0.1 and 3, so this big force. So the more water you put in a confined space, the more hydrostatic pressure you get, up to around 3 megapascals. When this hydrostatic pressure drops to 0, the plants start to wilt. All the support they're getting from this turgor pressure is reduced, and the plants start bending in on themselves, which we've seen in plants in dry times of year. What happens to a plant through periods of drought? Well, on the x-axis we see pure water here on the left, all the way down to a water potential of negative 4. and we see what activities takes place in a cell as the water potential drops. The first thing to stop is cell expansion. So as plants are growing, their cells expand less in dry times. And it takes a very small drop in water potential for cells to stop expanding. 50% of the expansion of a leaf is purely due to the influx of water. So if a plant has less water as leaves are being produced, their leaves will become smaller. And they do this for a reason, because if they're developing in dry times, they don't want really big leaves, because the bigger the leaves, the more water they have to use. So there's a reason one of the first things that stops during dry times is cell expansion. In line with a drop in cell expansion is a reduction in wall synthesis, so everything that's producing cells gradually starts stopping. Cellular function stops over a water potential deficit of -1. So we get ribosomal function or protein synthesis stopping. And around -2, photosynthesis and stomatoconductance, so movement of gas in and out of the stomata, stops. The stomata close at about a negative water potential of 2. As water potential drops even more, the plants start accumulating solutes in their cells, and that's to try and prevent or decrease the chemical energy inside the cells to stop water loss. And here we have the first reference to one of the most important hormones in plants, abscisic acid. As plants start suffering from a drought, they start producing abscisic acid, and this abscisic acid plays very important roles in the plant that we'll be going through in great detail in this subject and in later subjects. So as water potential starts dropping really low, abscisic acid starts being accumulated. That's the end of this topic. In the next topic, let's start building up this concept of water potential into understanding how water functions across an entire plant.